think you're in for a treat. A Wednesday afternoon lecture with a title like this one, Adventures in Brain Plasticity from Memory Palaces to Soul Cycle. Dr. Wendy Suzuki is going to walk us through, or maybe we'll be running through those various palaces and cycles. And it's a pleasure to have her here this afternoon as our Wednesday lecturer. So welcome to all of you and to all the people who are watching by video, which I suspect is quite a large number because that seems to be the way things go. But we have a lot of people here in Missouri as well, so glad to see all of you. Dr. Suzuki is currently professor of neuroscience and psychology at the Center for Neuroscience at New York University. And she has a career that includes an undergraduate degree at Berkeley, a PhD in neuroscience at UC San Diego, and we are happy to claim a part of her training also because she was here as a postdoctoral fellow in Building 49 in the laboratory of Dr. Robert D. Simone. From there, she went on to the current positions at NYU, where, as I mentioned, she is now a professor. I'm also happy to report, when she was a postdoc, got a FAIR Award. All of you postdocs who are going for those, look where it gets you. And she's also been recognized particularly for her teaching excellence, which I think we're about to have a chance to experience by the awarding of the Golden Dozen Teaching Award from NYU College of Arts and Sciences. She's also a NIH grantee, and thank you, Wendy has been a member of grant review study sections, which is an important part of being a citizen of our community. Her major research interest is in brain plasticity, but her work is especially well known for the work that she's done about how we recruit and retain long-term memories. And more recently, she has turned her attention to the question of how aerobic exercise can be used to improve learning, memory, and higher cognitive abilities, hence the reference to Soul Cycle. So please join me in welcoming Dr. Wendy Suzuki.
Thank you so much, Dr. Collins, for that wonderful introduction. It is such a pleasure and an honor to be here, to be able to give this talk. And what I wanted to try and do is give you an overview of the different topics that have fascinated me over my career in neuroscience. And the main theme is brain plasticity, which I define as the ability of the external environment to change something in the brain, the anatomy, the physiology, or the function of the brain. And I wanted to start kind of historically. How did I get interested in this topic in the first place? And it really had to do with like for many of us, a professor that oriented me to the wonders of neuroscience. Her name was Marian Diamond at UC Berkeley. Can you tell which one she is? So yes, this is Marian right in the center. And she and her colleagues, including Mark Rosenzweig here, in the late 1950s, early 1960s, were really the first ones to show an example of real brain plasticity. Now, I have to step back in it for a second and remind you that at this time period, late 1950s, early 1960s, all of us know that the brain can change very dynamically in adulthood. But at that time, the idea was there's no change in adult brains. Once you get to adulthood, no change at all, because they didn't have the technology to look at that, to be able to see that. And so what Marian Diamond and her colleagues did was a simple but very elegant experiment, one of the very first ones to show that the adult brain can change. And what they did is they raised rats in what you see here, what they called enriched environments, which is basically like the Disney World of rat cages with lots of other rats and lots of toys to play with. And they compared the brains of these rats to rats that are raised in impoverished environments and showed for the first time changes in cortical thickness, anatomical changes, changes in neurotransmitter levels, and changes in synaptic connections. So this was just fascinating. I learned this the very first day of my freshman year in her class, Brain and Its Potential, and that really oriented me, really for the rest of my career, to try and understand that level of plasticity at a deeper level. Well, when I got to graduate school myself, the form of brain plasticity that I wanted to take on was probably the most common form of brain plasticity that we experience every day. That is the ability to form and retain new memories, name-face association. So I met several new people here today, and that ability to associate that name with that particular face, that is a form of plasticity, a form of memory. And when you talk about memory, the classic study there is, of course, the study of a 1957 study published by Scoville and Milner of the very famous neurological amnesic patient, HM. We now know his name was Henry Mollison. And for those of you who aren't familiar with the study, HM had suffered from very severe epilepsy. And at that time, as today, one of the approaches to relieve that epilepsy was to remove the medial temporal lobe and a key structure called the hippocampus on one side of the brain. Well, this epilepsy was so severe, they decided to do an experimental procedure to remove both sides of this medial temporal lobe area focused on the hippocampus. And while it did improve his epilepsy symptoms, it left him with a lifelong, profound memory deficit. And I just want to show you in a different view the location of this hippocampus, which is going to be the focus of the first half of my talk. Is situated deep in the human temporal lobe. This is the front of the brain. Back of the brain, we have the temporal lobe cut out here. Hippocampus means seahorse, and this is my favorite illustration of why it's called a seahorse. This is a dissected human hippocampus, and here is a seahorse. So the classic studies of Scoville and Milner, and later studies, particularly by Brenda Milner and her colleagues, showed that, identified that the hippocampus was critical for memory, but not any form of memory, a very particular form of memory that is generally described as declarative memory, made up of two different forms of memory: episodic memory, that is memory for the episodes or events in our lives, complicated form of memory that includes all the smells and the feelings and the people and the places that are involved in the different episodes of your life, but also semantic memory, memory for facts. Mort Mishkin here at NIH has also called this form of memory cognitive memory. So while Brenda Milner and Scoville and their colleagues were the first to identify this structure, the hippocampus, as key for these key forms of cognitive or declarative memory, actually thousands of years before Scoville and Milner, people already had a very deep understanding of how to strengthen this exact form of memory. And that procedure was called the procedure of memory palaces. When I say ancient, I mean ancient. It was Simonides that has been given described as originator of this idea way back in 1556 BC. And the idea here was that they developed these approaches to be able to memorize whole sets of things. Remember, this is way before the time of printed books, way before the time of Google that you all know we use our memories for now. And so they really had to have a way to be able to memorize long poems, Bible verses. And the approach that they developed was to picture a spatial location you're very, very familiar with. If you live in a palace, it could be a palace, it could be your house. So start with that very familiar spatial location, and then take a visual or a virtual walk around that spatial location that you know, and then place the new items or the new phrases that you have to remember in a particular order in different spatial locations around the location. And by practicing this technique, you are able to memorize very, very long stretches of things. This form of memorization we know now is very dependent on the medial temporal lobe. Now, you might be familiar with this general idea, not from the concept of memory palaces, but perhaps from this book, a very famous book by Luria, who he described somebody with extraordinary memory, who he himself describe this kind of memory palace technique. The interesting thing is neither his patient, described in the book as patient S, or Luria himself was familiar with the ancient memory palace technique, but that's exactly what he was using. And to bring it even more contemporary, current competitors in memory competitions, including, you might have read this book, Moonwalking with Einstein, also use this memory palace technique. And I find it interesting because it really taps into some of the key functions that we know are important for, that the medial temporal lobe are critical for. First one is association. Why did they have people walk around a space that they knew very well? Because they realized that the well-formed memories of those spatial locations could actually pull in the new memories very easily. So something that was being worked was the associative memory. They recommended that you use very vivid imagery if you want to remember these lists of things. And so they knew that emotional resonance was important for memory. We now know that is because of the interconnections between the amygdala, key structure for emotional memory, and the hippocampus. They focused on spatial organization. We know that the hippocampus is critical for and very good at remembering spatial information. Of course, this was not known at that time. And finally, they used temporal order to be able to memorize things in a particular order. That's also something important for the medial temporal lobe. So I start with this introduction because what I want to try and do in the first part of the talk is to focus on two of the four key aspects that are mediated by these medial temporal lobe structures. I'm going to be talking about the role of the medial temporal lobe, specifically the role of the hippocampus in formation of new associations. How does it do that? Why is it so good? Is it good? What is the evidence? And in the formation of memory for temporal order. So now we're at 1998. I have finished my wonderful postdoc here at NIH, and I get a faculty position at New York University. And the approach that I want to use to start to study these things is single unit physiology in awake behaving primates. Because what I really want to do is go into the hippocampus and record the activity of individual cells as animals are either making new associations or forming new temporal order memories, and ask what is actually happening in these structures. So these I'm going to go over these two key results, starting with associative memory. So first, let me introduce you to the species we're going to be focused on. This is a ventral view of the macaque monkey brain. And outlined here are some of these key structures of the medial temporal lobe that surround the hippocampus. This is the entorhinal cortex here, perirhinal cortex, parahippocampal cortex. We're looking at the ventral view of the monkey brain. And the key structure I'll focus on today is the hippocampus itself, and the monkey is situated just deep to these cortical areas here. And so the strategy is to find a simple yet powerful task that requires the animals to form new associative memory and record individual cells in the hippocampus as the animal is doing this kind of dynamic memory formation. And here is the task that that we chose to look at new associative formation. It's basically a version of a name-face association that you would do in people. Instead, we're having them associate a particular rewarded location with a complex visual scene. So this is what animals had to do every day. Sitting in front of a video monitor, here's a representation of the video monitor. They have to fixate to the central fixation point. Then we show them a big complex image that fills the entire computer screen. Superimposed on this image that you could just barely see are 4 targets, white targets, north, south, east, and west. They have to maintain fixation for 500 milliseconds. And then there's a delay interval, what they have to continue to maintain fixation, and the scene disappears, but these targets remain on the screen. And then the animal's cue to make a response is when the fixation point disappears, which is the animal's cue to make an eye movement to one of the 4 targets, and the arrow is just a representation of a possible eye movement the animal could make. Now, at the beginning of the day, of the training session, the animal doesn't know which target is associated with reward. So he makes errors, and with trial and error, he will eventually learn for 1 to 4 new location scene associations which target will get him a big fat drop of fruit juice when a particular visual scene is shown. So we have the animals doing these tasks. They're learning 1 to 4 new associations during a recording session. We are recording individual hippocampal cells. And first, I just want to show you generally how these hippocampal cells are responding to this task. And here what I've done is I'm showing you the activity in spikes per second, and here is the time course of the entire trial. During the baseline, here's the scene presentation, delay, and eye movement, and I'm averaged together all the trials in which 3 different scenes are shown. This red response is a response to when the gorilla scene was shown. And the green and the blue are 2 other scenes, just to show you that hippocampal cells in this task really differentiate between the visual images. We call this visual selectivity. They're responding very selectively. But that's not the question we wanted to ask. We wanted to ask, as the animals are learning the new scenes, because they don't know it at first and they learn it, how do the hippocampal cells respond during that formation of a new association? To ask that question, we need to look at the animal's behavioral learning curve for a particular scene. So here is a beautiful learning curve that the animal exhibited to scene 653. This is probability correct, 100% here, 20% here, chance is 25%. And I'm showing you a dynamic logistic regression algorithm estimating the trial by trial probability correct over these 55 trials. And I'm just going to show you what the raw data look like up here. The red dots for each trial are the error trials. The blue dots are the correct trials. So clearly, there is a period, about 18 to 20 trials, the animal's performing very poorly. There is a very steep learning curve, and then there's very good performance after that. What are hippocampal cells doing here, here, and here? So let me superimpose, using this axis, an example hippocampal cell that we recorded from. I've subtracted away activity during baseline, so that's 0. And what I'm showing you is the average trial during the delay period of the task for the first 18 trials. What are these hippocampal neurons, or what is this hippocampal neuron doing during the first 18 trials when clearly behavior is very poor? He does not know this association. What we see is activity a little bit above baseline, but no change across these 18 trials. The critical question, what happens here when there's clear and dramatic behavioral learning? What we see is, in parallel with this clear and dramatic behavioral learning, a clear and dramatic change in the neuron's response properties. These cells are going up in parallel with learning. This one is following just a little bit from learning, and that activity is staying up for as long as we're able to hold the cell. Now, with all the control trials we did, I can assure you that this change in activity is only happening for this particular scene, not for the other scenes, including if the animal learned it, and not for another visual scene that was well learned, in which the animal is making the same rewarded motor response and getting the same kind of reward. These are not a reward-related activity, but it really seems to be an activity specific for learning a particular association. We found overall about 18% of all the cells in the hippocampus that signal this kind of activity, and we call these cells baseline sustained changing cells, or for short, changing cells. So the next question is, well, what does this change in activity represent? It's just an increase in firing rate. What we wanted to look at is how selective these cells became over the learning process. Remember, selectivity is you're highly selective if you only respond to one particular visual item or one item. And you're not selective if you don't respond at all or respond similarly to lots of different kinds of visual items. And so what we found is all of these changing cells in this category are increasing their selectivity after learning relative to before learning. So all these hippocampal cells are kind of tuning their selectivity, becoming more selective to those learned associations. And to give you a better feel for it, I can show you the raw data. So this is response in spikes per second over lots of all the trials for a particular training day. And these different colors are the different scenes. 2 of them are newly learned scenes that the animal's learning at the same time. This white one is one of the new scenes, and the green one is also a learned scene. That one didn't change. And then the other ones are associations that are very well known to the animal, not changing here. But you just get a feel for early on, there's no differentiation. These cells are not differentiating. And then the cell becomes highly selective to a particular learned association. And I'll just add the learning curve here. So the take-home from these set of studies was that we asked the question, why is the hippocampus so good at associating things together, either old things with new things coming in? In this case, it's associating 2 new things together. It's because we think that the hippocampal cells participate in signaling new associations by changing their stimulus-selective response properties. And there's a good number of these hippocampal cells that are making this shift, kind of tuning their response properties as new learning comes in. So this gave us kind of first insight into how these hippocampal cells change, and this work was done by the postdoc in the lab at the time, Sylvia Worth. So that was part of our foray into association and why the medial temporal lobe and the hippocampus in particular is so important for association. Now I want to turn my attention to temporal order. Now, in these memory palaces, you are walking through these familiar places, and you're placing the items you want to remember, the phrases you want to remember in spatial locations along the path. But it also helps you remember the ordering of these things. When there's a poem, you have to remember the order as well. So what is the role of the hippocampus in temporal order? Again, same strategy. Let's use a simplified task that requires the animal to use this form of memory and record in the hippocampus and see what we get. So this task is a temporal order memory task. There are 2 phases of the task. The encoding phase, animals fixated a central fixation spot here. Animals were shown a first image here and just had to maintain fixation. There's a short delay, maintain fixation. A second unique object is shown as the second cue. And in this task, they have to remember what was shown and when it was shown. Continue fixating, and as long as you fixate through this encoding phase, you get a small reward. After a little break, you move into the retrieval phase, where after delay interval, we're going to show you the first and the second item shown in a random position on the screen, along with a distractor item, the pumpkin. And what the animal has to do to get the trial right is first touch the image that was shown as the first cue. That's the sunflower. That will disappear if he does it correctly. And then touch the item that was shown as the second cue. Stay away from the pumpkin. If you do that, if he does that well, you get a very large juice reward. So we had animals trained up on this, and we wanted to ask, how does the hippocampus signal the encoding of, how does the hippocampus encode new memory for temporal order? And so, again, to perform this task, you have to know what was shown and when it was shown. So the first thing we ask is, how many hippocampal cells are simply differentiating between the different items that are being shown as cues? And there were 8 possible items shown over here. And remember, we had a lot of hippocampal cells that were differentiating between visual images on this other task. So we were surprised, here's the hippocampal response, when there were so few cells responding selectively to these items. I'm not going to talk about these other medial temporal lobe areas, but you can see clearly these other areas are the areas where the object was, or the item was being differentiated, but not in the hippocampus. Okay, we thought, well, if they're not distinguishing between different items, maybe they're keeping track of trial timing. That is, whether it's the Q1 period or the Q2 period. I don't care what's being shown there, but can you differentiate between whether you're in the Q1 period or the Q2 period? And when we asked that question, what we found was a huge number of cells. Here was that item response down here. Here was the huge proportion of hippocampal cells that are responding and differentiating between whether you're in the Q1 period or the Q2 period. So what do those cells look like? So here are two examples of what we called hippocampal time cells. An example here, example here. Here's spikes per second on the y-axis, and on the x-axis is the time course of the encoding period. Here's the Q1 period, that delay period, the Q2 period. Look at the interesting thing that's happening. In this cell, no response during Q1, but look at this kind of ramping up of activity that we saw, and high activity during Q1. This other cell responded a lot during Q1 and ramped down its activity to Q2. So this is what we found when we dug deeply. What does this actually represent? So that was the key question. To address this question, my very talented postdoc at the time, Eugene Naya, decided to look at the population of all the hippocampal cells recorded at the same time, because they're all recorded in the same exact time frame. And what he did is he created an n-dimensional vector with the responses of all the hippocampal cells as the different dimensions of the vector. And he first chose a template period. What is this big vector doing during template, which is the Q1 period? And then what he did is he took 40 millisecond slices of mini templates all along the delay interval between Q1 and Q2 and asked how similar was this vector early in the delay to this template of Q1. And then what about the next 40 milliseconds, the next 40 milliseconds, and he all the way through the delay interval. Let's see what the population does. What he found was early in the delay interval, when you're very close to that, so the Q1 period is right here, the vector has a very similar response pattern. This is a measure of distance, so low distances, similar kinds of responses to the target period. But look at what's happening. As you go through the delay period, the whole vector is becoming more and more different in a systematically increasing way. So the distance of the whole vector from this early period towards the late period is giving you kind of an incremental timing signal. Well, what if we look at it in a different way? Let's use now the template as not Q1, but the Q2 period. And let's look at the full population of what the hippocampus is doing during this delay interval. When I use the Q2 period as the template, here's what I find. So this is the beginning of the delay, this is the end of the delay. Far away from the Q2 template, you have the biggest difference in the hippocampal population, and you gradually and incrementally get closer. What does this mean? This means that the cells in the hippocampus as a population, and we later confirmed that individual hippocampal cells can do this, give what we call an incremental timing signal. It's telling you where in this critical delay period you are when you're having to pay attention to and differentiate between the first Q presentation and a second Q presentation. And in fact, this was very exciting because the same time that our paper came out in the monkey, Howard Eichenbaum came out with a parallel study where he identified what he called time cells in the rodent hippocampus that are a little bit different, same kind of flavor, but a little bit different. These cells, over a longer delay interval, my delay interval was about 1 second long, his was more like 20 seconds long, he found individual hippocampal cells that would only respond at the very beginning of this delay interval or at the 5-second mark of the delay period every single time the animal did this delay-like task. And so they were time markers, different flavor. And so, in fact, right now, we are collaborating with the same computational modeler who has looked at the rodent data, and he's now taking the monkey data to ask, how can we look at the similarities, or what are the similarities and differences in these prominent timing signals that we're seeing across the rodent and the monkey hippocampus? I should just say that this is so exciting because for so long we knew the hippocampus was critical for episodic memory. A key aspect of episodic memory is that it has an ordering. It has a beginning, a middle, and an end. And so what we're seeing here are two ways in which the hippocampus has the capacity to create that kind of episodic memory. That is, there's background activity that is keeping track of incremental timing, and associative, things that are perhaps able to link particular items or events to particular time periods within a particular episode. So that's how we're thinking about how this might be expanded to our more complicated understanding of episodic memory. So the take-home from the hippocampal work is that these hippocampal cells provide a prominent incremental timing signal that may provide ongoing information about the temporal context of particular encoded episodes. So that was 25 years of work, fascinating work, in one realm of brain plasticity, looking at memory mechanisms in the medial temporal lobe, basic studies looking at this. And now I want to turn to the new area of research that I've studied. And this was not inspired by a professor or an article that I read, but instead this was a much more kind of personal decision to look at something different. And this started when I was getting tenure at New York University, working really, really hard, that I know all of you know how to do here at NIH, but it's a six-year slog to get tenure, and you are so engrossed in all of this. And the science was going great. Lots of exciting people, lots of exciting findings, but nothing else was going particularly great. Gained 25 pounds, did nothing but work in the lab, and I definitely needed something to rebalance my life, and I happened to find something that could help me when I went to the gym. So, obvious thing, gained a lot of weight, go to the gym. So I went to the gym, really got active, and I found this workout that was very, very inspiring. And it was developed in New York City. It's called Intensati. And you go into class, and it's a combination of physical movements and kickbox and dance and yoga and martial arts. And but on top of that, with each move, you yell out a positive spoken affirmation. And it is ridiculous the first time you do this class because you feel like a complete idiot. But at the end of the class, you come out feeling just like $1 million, because exercise, as we know, improves your mood, and if you yell out positive affirmations at the same time, it kind of boosts that effect. So lost weight, got much stronger, noticed an immediate improvement in my mood. But the thing that really struck me happened about a year and a half in to this new exercise regime. All the monkey studies were going fine. And this happened one day when I was sitting at my desk writing an NIH grant. I know I'm not talking to the right audience to get a lot of sympathy to writing an NIH grant, but you could imagine. And the comment that I made to myself that really took me by surprise was the following. I said to myself, gee, writing is going well today. That was really striking because writing an NIH grant never went well. It was stressful, it was, pull your hair out. But that day, I noticed that writing was going very well. And when I thought a little bit deeper about it, I noticed that it was going well because I was able to focus my attention deeper and longer. And not only that, my memory, my declarative memory for all those little details from all the different papers that I was using to synthesize my award-winning thesis for my grant, that memory was better too. And that really got me thinking. And the thing that happened was, when you're a professor at a undergraduate institution, and you want to learn a new topic, like the effects of exercise on the brain, the first thing that you do is you decide to teach a new undergraduate class on it. So I decided to teach a new undergraduate class called Can Exercise Change Your Brain? Literally the question I was asking myself. And I went through and I read the literature, and I found 3 key findings. 1, what about mood? We know from studies in rodents that exercise significantly increases levels of serotonin, noradrenaline, dopamine, and endorphins during and after the workout. So that can help explain the mood boost that I got every single time I went to the gym. I went to the human exercise literature, and the most common finding is that either a single bout or longer-term exercise improves your ability to shift and focus attention. That's what I was noticing in myself, too. And third, memory effects. I'll get back to that. But memory effects, we know that increased aerobic exercise, including all the beautiful work done here at NIH by people like Heather Cameron's group, are showing that exercise and including enriched environments can stimulate neurogenesis, the birth of brand new neurons in the hippocampus, one of only 2 brain areas where new neurons can be born in the adult brain. And I thought, well, maybe that's what's happening to me. I have more neurons in my hippocampus. So I decided to teach this class, and because I was inspired to go to the gym, going to the gym inspired this class, I decided I wanted to bring exercise into the classroom. And I wanted to have my students actually exercise, and then I'll teach them what exercise is doing to the brain. So I went to my administration, and I said, I have this great idea. All you have to do is pay for an exercise instructor, and they'll teach the exercise, and then I will teach the academic part of the class. And they said, great idea, but we pay you to teach, so there's no extra money, so no, you can't do that. And so I went back to my office, and I decided to do the next most obvious thing. I decided to go to the gym and become trained to teach the exercise part of the class myself. So this started the most extensive class preparation that I've ever had in my entire career. I went to teacher training, and then I trained to actually teach an exercise class, this intensati class, to my students. And of course, I was preparing the academic part of the class as well. And I just want to bring you back very quickly to that first day of class, which was September 7th, 2009. And there were 3 things that were very, very different that day. The first thing is that I strode into class clad head to toe in my best Lululemon because I was teaching exercise. It's a little bit different. Number 2 was I was nervous. And I'm very comfortable speaking. This was, I had 12 years of undergraduate teaching under my belt, but I'd never taught the exercise class, so that I remember that. The third thing that was different was the students themselves. So they are, they're usually excited, maybe a little nervous the very first day of class if they don't know what the class is. These students look scared. They did not know what, and they saw me in my Lululemon, that really scared them. And so I knew that I wouldn't, be able to, the only way to know whether it's going to work is to stand up there and actually teach the class. And I have to tell you that shifted the classroom so much. And it also shifts a lecture hall so much. So I'm going to ask you all to stand up for 2 minutes, including the people watching. I see you. Okay, so this is very, very simple, and this is going to make you remember everything that I've told you today. So this workout is called Intensati. All you have to do is do what I do and say what I say, and I don't have music today, so you just have to listen to me, okay? So first thing we're going to do is punches right and left. It is 5, 6, 7, 8, right, left, right, left, right, left, right. I say, I am strong now. You say it. Again, I am Superman strong. You say it.
I am Superman strong.
I am Wonder Woman strong. Your turn.
I am Wonder Woman strong.
New move. Uppercuts. Right and left. Right, left, right, left. I am inspired now. You say it.
I am inspired now.
One more time. I am inspired right now. You say it.
I am inspired right now.
Okay. Have a seat. Okay. So I want you to think back and ask yourself, what would it be like if I got to do that for a whole hour at the beginning of every college class that I ever took, taught by the instructor? And I can tell you that it completely changed not only the way I teach the class, because the level of engagement and interaction that I got by first working everybody out and then going into lecture was transformative. But the other way it was transformative was that it made me really think twice about how fascinating the effects of exercise on the brain was. But it takes a lot to actually shift your research program. And I have to say that at the same time that I was noticing in myself that my memory, my declarative memory was getting better, my attention was better, my mood was a lot better, was the same time that my father started to suffer from dementia. And it was a very precipitous drop. And I noticed that all the things that were improving in me, they declined clearly in him. And I thought, he's 85. He hasn't been to one of my intensati classes, sadly. But what would have been different if he had exercised leading up to his older age? So all these questions going through my mind. And that's what led to these new findings. And I just have to say that these findings were clearly originated with some of the beautiful work that Marion Diamond first did, because she showed, as I said, environmental enrichment changes the brain. She showed changes in cortical thickness, in neurotransmitter levels, synapses, angiogenesis growth factors, hippocampal neurogenesis, as I mentioned. But then later studies, in particular by Henriette von Prague, asked what was it about, oops, I wasn't supposed to go, what was it about this rat-enriched environment that was causing these changes? And she found that it was exercise. Exercise is causing the majority of all of these anatomical, physiological changes, and not just anatomical and physiological, but increased aerobic exercise is improving a widening range of hippocampal memory tasks in animals. Okay, so what does the human exercise literature tell us? Well, first, I use this picture because the vast majority of studies on the effects of exercise have been done in the elderly population, 65 or older. And the second biggest group is children, and this big group includes me, non-aged adults, that I was seeing such big effects. No data there, and so that's a big problem. I became familiar with all the effects of aerobic exercise and hippocampal neurogenesis, hippocampal function. Went to the literature. Where are the hippocampal improvements? Not there. Interesting. Maybe it's because started to develop a hypothesis. Maybe it's because the older people are not exercising to a high enough level. Maybe we need to start doing some studies in healthy young adults. And finally, in this exercise literature, what is the mechanism? This is a big question. Very little information about that. So these became the unifying questions of my human exercise work, looking at, particularly interested in effects of exercise in non-aged adults, but leading up to aging, asking when or if we can see improvements in hippocampal function, as has been shown so beautifully in the rodent studies, and starting to think seriously about mechanisms. So the two key goals of this new program of research that I've started at my lab at NYU are, one, to define the exercise prescription for optimal brain function. Depending on your age, your genetic background, your fitness level, what is that prescription that is best for you for the range of cognitive functions that can be improved? And second, to delineate the pathways and mechanisms that are being used to enact these effects. And I'll just highlight the role of a wonderful postdoc in my lab doing the exercise work, Dr. Julia Basso, and Kristen Krosta, a great research associate. Two categories of studies that we've done are immediate studies on the effects of exercise on brain and cognitive functions, and long-term studies. So let me just quickly give you a flavor of these kinds of studies that I've done. So the first is we were looking at one bout of indoor cycling, 1 hour of indoor cycling, and comparing the effects on prefrontal function right after the workout versus hippocampal function. So here is the experimental design. We did cognitive testing. We had them do an hour of indoor cycling. We had different delays, but I'm going to show you all the data combined in one. And we cognitive tested after these various delays. And while this group actually did the exercise, the control group watched somebody else jump around. So they watched the TV show 24 for the same amount of time. And after delay interval, we tested them cognitively. And so these were healthy NYU undergraduates between the ages of 18 and 35. So we had a whole wide range of tests. We focused on the attention task because, as I said, this has been shown most in the literature, but not all the tasks at the same time. So we wanted to have a range and look at lots of different tasks, as well as a few of hippocampal long-term memory tasks, including Hopkins verbal learning tests with longer delays and this visual recognition memory test. Benton visual recognition memory test. So here are our results. Prefrontal cortex, what you see on all of the prefrontal tasks, Z-scored together, what you see is here's the video watching group. You improve the second time after the intervention because you've taken the test before, so you get better, but you improve significantly more on average on all the prefrontal tests after a bout of acute exercise. This means that every time you go biking for an hour, that you have this attention improvement that lasts somewhere between 30 minutes and 2 hours. By contrast, there was no difference in the performance on hippocampal tests after the intervention, although this at some point was significant, which is, notice, this is the video watching group getting worse at hippocampal tests. At some point, we thought that the title of our paper would be watching the television show 24 is significantly impairing to your hippocampal performance. This was not significant, but we're actually interested in going back and doing this again with a larger number of people and with a larger set of delay intervals. Okay, but really, the studies that I really wanted to do when I started this work was long-term studies. What if I can get you to increase your cardiorespiratory function over 3 months, increasing your cardiorespiratory output, and then can I test your attention, your memory, your mood? And so we've teamed with a wonderful indoor cycling company called Swerve, not SoulCycle, but Swerve. And here is our experimental design. This is preliminary data. We're about halfway through, a little bit more than halfway. We have between 15 and 20 people completed in this design. We test them on a whole wide range of tests. These are low-fit people between the ages of 35 and 59. Low-fit people, they start to get free exercise classes at a beautiful studio, and we ask them to go 3 times a week for 12 weeks, and then we bring them in again and do cognitive testing. Now, the thing I have to tell you about Swerve is that when you go in to take the classes, you join the red team, the blue team, and the green team. And this company actually has a way to estimate your caloric output. So they estimate your caloric output of everybody on the green team, average it together, and put it on the screen at the front of the room and say, green team, this is your score. But you're lagging behind the red team, so cycle more. So it's a fun kind of way to get you to, without pointing you out, Wendy Suzuki on bike 21, you're not cycling very fast. So it's a much more positive way. So we wanted to match that in our control group. So while this is Swerve classes, we had competitive video game playing as our control. So our control subjects come in and they have a rousing video game competition with the undergraduates in my lab. Every time they come in 3 times a week. They get all the kind of attention that the other group has, and we test them before and after. And here's what we found so far. So we had a wide range of mood measurements. What is this doing to overall mood? And happy to see that 3 months of increased exercise significantly improves positive affect in the exercise group, but not in the video game group. And moreover, we see a significant relationship such that across all of the participants, such that higher fitness gains, the more you're changing your VO2 max, your cardiorespiratory function, the greater improvements in mood that you experience. Okay, what about hippocampal dependent memory tasks. This is what I have been fascinated with since I started to get interested in this subject. And we are using a mnemonic similarity task. This task, we chose it very carefully because we know that it activates the part of the hippocampus where neurogenesis takes place. We suspect that the same kind of neurogenesis is happening in humans as it is in rodents. And this is a task that we know is engaging that part of the hippocampus. So what subjects do is they look at an original image, and then there's a surprise second portion where they are shown either the same image, and they have to say it's old, a similar image, and they have to say it's similar, or a brand new image. And again, this task engages and is sensitive to this area that is presumably undergoing hippocampal neurogenesis. And what we found in our preliminary studies is significant improvement in the exercise group on the recognition portion of this task relative to the video game watching group. So still work in progress, but one of the first demonstrations of improvement of hippocampal function following long-term increases in exercise where we can confirm that cardiorespiratory function has improved. Okay, so let me just, in the last minute, go over some of the ideas for pathways and mechanisms. And they really center on a growth factor that many of you are familiar with, brain-derived neurotrophic factor. And the issue there is from the rodent studies, we know this growth factor is definitely involved in hippocampal neurogenesis. The problem for the human studies is peripheral measurements of BDNF levels are contaminated with all the other roles that BDNF have. So it also plays a critical role in vascular health, for example. But very excitingly, there are 3 recent papers that show a second or 3 additional peripheral factors that may be much more related to central BDNF levels and stimulation than just peripheral measures of BDNF itself. The first is irisin, that was identified by Bruce Spiegelman's lab at Harvard. It is a hormone that is released with exercise, passes the blood-brain barrier, and presumably stimulates BDNF. The second is identified by my colleague Moses Chow at NYU Medical Center, beta-hydroxybutyrate, a ketone body released by the liver with exercise, passes the blood-brain barrier, and stimulates BDNF translation in the hippocampus. And third is cathepsin B, or CTSB, identified by a collaborator, a colleague of mine, Henriette von Prague at NIA in Baltimore, which is a myokine released by muscles that are exercising, also passes the blood-brain barrier, and if you knock it out in rats, they do not benefit from the effects of exercise on hippocampal function. So several of these, irisin and CTSB, has been shown to increase in people exercising, but nobody has looked at this in relation to changes in cognitive function. And so these are some of the mechanisms that, or some of the molecules, I should say, that we're starting to look at in people that have well-characterized changes in exercise and changes in cognitive function. And so what I want to do is just end with the implications of this work that really make it so fascinating to me. And the first implication is one of the reasons why I got into the work in the first place. So I noticed how much exercise is improving myself. What are the implications for education? What if we integrate in a scientifically rigorous way exercise into education? Well, it turns out that I am at the largest private university in the country, NYU. And this is a picture of NYU's graduation. Yankee Stadium is the only venue large enough for the graduation for all the students and all the parents. So we've started a pilot program with freshmen looking at the effects of 1 semester on exercise, 1 semester off exercise, to try and take advantage of the resources that I have and look at the effects of exercise, not only on the cognitive functions that I've been talking about, but on study habits, drug use, even academic performance. And so those studies are in the middle right now. As I mentioned, aging, critical issue, and we are applying to NIA for some of these grants to look at the effects of exercise in a younger and an older population to start to see whether there are differences and sensitive periods across the aging before you get up to 65 or 70 years old. And finally, neurological disease states. We're particularly interested in ADHD and autism. And we've actually done work in traumatic brain injury. So all of these kind of aspirational kinds of questions. So this was the full adventure in brain plasticity from memory palaces to soulcycle. And I just want to end with a very practical note, because at the end of my talks, everybody says, okay, that's fine, Wendy, but just tell me how much I have to exercise. But just give me the formula. Actually, and then they say, oh, wait a second, tell me the least amount of exercise that I have to do to get any of those effects that you have. And so the good news is that it looks like you don't have to become a triathlete to get these effects. But on the other hand, if you're a walker, that's great. Walking is great, but you're not going to get all of the benefits. So something in between walking and being a triathlete. And I think the exciting thing is, sorry, that's a little bit vague, but that's where we are. And I think the exciting thing is that these different cognitive functions are going to have different formulas. So we already know that mood is improved by simple walking. It's also improved by aerobic exercise, but you can get that with simple walking. I don't think you can get the hippocampal effects with just walking. And I'm not sure what the minimum level or the range of the optimal level is for the attentional effects that would be beneficial not only to students, but to researchers working in the lab, as well as to the aging population, given the age-dependent effects on both prefrontal function and hippocampal function. So with that, I would be happy to answer any questions, and I thank you for your attention.
Thanks, Wendy. That was inspiring, and it did get our blood flowing, and that's always good on Wednesday afternoon. There are going to be questions, and the mics in the aisle are there so that people listening by video can hear, and maybe we'll start right over here.
Hi. When you were randomizing your subjects
Yes.
to exercise and control, did you do that before or after you did the cognitive pretest?
Before.
Okay.
It was all done before, yes.
Okay. We'll just alternate back and forth over here.
Great talk. As a long-time person who's enjoyed fitness, such as marathoning, see I'm wearing a marathon shirt here today. I'm curious, those tests that you did that showed improvements in these different cognitive skills.
Yes.
How long, I'm sorry, I'm having trouble formulating the question. If you continue to repeat that, will they continue to get better? Before, or is there a point at which that starts to plateau and they no longer improve?
Yeah, that's a great question. And the answer is we don't know the answer to that. And I mean, I've always been, one of the many experiments I want to do is look at, for example, Olympic athletes. So these are people that are really exercising to a high level. Have they maxed out? Can you see a big improvement when they first start their training? And all of those time courses are almost not studied at all. There's a single time cohort, including our first studies. As we get more funding to do this, those are exactly the questions that we want to answer.
Great. Over here.
Lovely talk. I really enjoyed it. So I was wondering if you have you looked at the sleep after exercise?
Yes, that is a great question, because one of the side effects, positive side effects of exercise is that you sleep better, and for the long-term studies, are these effects due to improved sleep relative to any change in the exercise regime that they're doing? And we haven't looked at sleep yet, but that is definitely on kind of the docket to look at. And it's easy enough to do. Actually, that's not true. We do have sleep survey questions on in the test that we do, but we would love to get some electrode company that's monitoring sleep in a serious way and get them to start using that for the duration of the study.
Just give them a Fitbit. It's pretty good.
Yeah, okay.
I have another question.
Yeah.
So, what's your personal opinion about sleep and memory retention? Is it for normalizing or renormalizing those synapses during when we sleep?
Yeah, I mean, I think there's beautiful studies in the rodent hippocampus that is showing that sleep is changing synaptic activity. It is consolidating new memories, and there's even some evidence that kind of consolidation is happening in people. I particularly like Matthew Walker's work at UC Berkeley, who is probably the best one, or my favorite author in that area. So yes, I think there is good evidence that hippocampal consolidation is happening during sleep.
Great. Next question.
That would have certainly livened up my undergraduate morning, so thank you.
You're welcome.
So my question was, you talked a little bit about the mechanisms of just in terms of hormones, but how do you think those hormones are then affecting the synapses directly? Are they working with long-term potentiation or some other mechanism, or do you know?
Yeah, no, I don't know. And that is where, I went from single unit monkey studies where I could be more direct in these questions, and I moved to this because the results that we have can be applied today, but I can't ask those deep questions. So one of the things and one of the strategies is to collaborate with rodent labs that are doing the exercise, the sleep studies, and the neurogenesis studies to try and answer those kinds of questions are much better answered in rodent or rodent models. Yeah.
We got time for two more questions, one over here and one over here. Go ahead over here.
Wendy, that was a fascinating talk. I was wondering whether the spatial role of the hippocampus, at least in the rodent, suggests that maybe, or has anyone looked at whether exercise through space, as opposed to on a stationary bike or a treadmill, or even exercise where you're changing directions but in the same locale, would have some added benefit, some interaction effect.
Yeah, you bring up a really fascinating point, and that relates to a study that Eleanor McGuire did in England, where she showed that cab drivers that use their spatial map for years and years and years and pass this really difficult test actually have increases in their posterior hippocampus. And one of the strategies that I'm interested in is combining the two. What if you do both exercise and spatial tests and make them do a lot of spatial mazes? Can you up the oomph of the hippocampus, given how much we want to preserve our hippocampus during aging? I think that's a really interesting question. I haven't done it yet, but that's definitely on the docket.
Great. Last question.
I have two questions.
How about just one, because I know people are anxious to go to the reception. Thank you.
What do you think the role of exercise is in patients with dementia? Is there data out there showing that delays things or makes things better for them?
What the data show are that exercise, even in early dementia, can improve cognitive function. So as long as you can actually do the exercise, you get benefits there. And so that's absolutely a direction. And that's part of the reason why so many of the exercise studies have been done in the elderly population, including in dementia patients.
Right. Well, if people want to continue the conversation, there's a reception about to get started in the medical library right around the corner. Please come and join for coffee and cookies. And meanwhile, please let's thank our speaker again.